So this is Cynthia Mwindachabe, and I'm representing Group 9 from Zamra. So this presentation was done by myself, Mufaweli, and Prudence Musonda. So ours was to look at biosimilars and their relation to biological reference products. So in the past weeks, we have had discussions on biosimilars, so I will not labor much to talk of what biosimilars are and biological reference products. So our task was to critically assess dossiers for the originator product, that is Remicade, and its biosimilar, which is Flixabi, as they relate to pharmacokinetics, human pharmacokinetics, clinical efficacy, and adverse events. So Remicade and Flixabi both contain the active substance infliximab. So infliximab is a medical product containing infliximab, which is an immunoglobulin monoclonal antibody. The reference product is Remicade, which has the same substance, active substance, and was registered on the market in 1999. So Flixabi has been developed as a biosimilar medicinal product to the reference product Remicade. So the first task we had was to identify a specific indication such as rheumatoid arthritis and quote specific clinical trial data or evidence from the EPA during our assessment. So from the information we gathered, infliximab is indicated for rheumatoid arthritis, adult Crohn's disease, pediatric Crohn's disease, ulcerative colitis, pediatric ulcerative colitis, ankylosing spondylitis, psoriatic arthritis, and psoriasis. So for the indication rheumatoid arthritis, we noted that 2 clinical trials were submitted to start with. So the first one was a phase 1 clinical trial that was conducted in healthy German subjects. So this one was done to compare the PK, the safety, the tolerability, and immunogenicity of Flixabi and Remicade. However, since this assignment was for us to look at a clinical study that was going to support the indication such as rheumatoid arthritis, we picked on the second clinical trial study that was provided, which was clinical trial phase 3 that was conducted in rheumatoid arthritis subjects. So this one was also conducted in subjects from UK, Bulgaria, Poland, just to mention a few. It was done to check the safety, efficacy, immunogenicity, and PK parameters. So the main aim for this one was to just demonstrate the equivalence between Flixabi and Remicade. So to answer the first question, it was evidenced through the second trial, which I just mentioned, which is phase 3 clinical trial. It was evidenced through trial number SB2G31RA. This was a randomized double-blind parallel group multi-center clinical study to evaluate the efficacy, safety, PK, and immunogenicity of Flixabi compared to Remicade in subjects with moderate to severe RA despite methotrexate therapy. So apart from that, it was noted that no any other clinical trial was conducted to cover for the other indications that were put up in the product information for Flixabi. So for these ones, we noted to say there was extrapolation that was supported by TNF alpha binding assay and cell-based assay. That is for the rest of the other indications. So the other task was to reflect on how we would manage the licensing of the biosimilar in all indications that were licensed by the originator, Remicade. So we discussed to say licensing for the biosimilar Flixabi in all the other indications that have been licensed for the comparator will be based on the establishment of biosimilarity between the two products. Then it would be justifiable to extrapolate the equivalent clinical efficacy and comparable safety profile from the Flixabi study in rheumatoid arthritis patients to all other indications where Remicade has been approved. So from here, my colleague will take over.
Good afternoon, everyone. The next task, we were requested to consult the EPA and at the end to find an appendix that had a divergent opinion from some CHMP members. Then we were requested to summarize the arguments in a slide. So some members of the CHMP did not agree with the decision on recommending the grant of marketing authorization for Flixabi. And their argument was that biosimilarity to Remicade was not established due to the following reasons. They had said Flixabi appeared to be associated with a higher incidence of anti-drug antibody than the originator Remicade. In a phase 3 trial conducted in patients with rheumatoid arthritis, the efficacy of Flixabi was constantly estimated to be lower than that of Remicade. The efficacy was significantly lower in ADA-positive patients than in ADA-negative patients. Also, patients with rheumatoid arthritis investigated in the phase 3 trial were treated concomitantly with immunomodulator therapy and could have exhibited less immunogenicity than patients in other infliximab licensed indications. As a result, the consequences of the difference in ADA incidences and the impact on efficacy in these indications was unclear. So the proposal by the applicant to resolve the concerns related to immunogenicity in the post-marketing setting by initiating a prospective observational cohort study in the indications of ankylosing spondylitis and Crohn's disease is considered inadequate. Then in addition, it is questionable to what extent a non-randomized observational study can provide data that will effectively address the uncertainties. So the second task, we were requested to check in the EPA. EMA works with 2 rapporteurs from 2 different agencies who take responsibility for the entire process. So we were requested to note out the 2 rapporteurs from the EPA. So the rapporteurs, we noted that there was Jan Muller Vegas from Paul Ehrlich Institute, German Federal Agency, and the co-rapporteur was Jens Hirstenberg from the Danish Medicines Agency. Then from these rapporteurs, Jens Hirstenberg was among the members that signed the divergent opinion. We were requested to discuss critically the arguments and form our own opinion from the EPA. And then what we noted was that the historical reported rates of ADA formation of Remicade have been around 8% in patients with rheumatoid arthritis treated concomitantly with methotrexate. The rates observed in both Remicade and Flixabi treated patients with the submitted RA trial were considerably higher. They had mentioned 5 to 6-fold, which was attributed to assay sensitivity in the current study. However, the applicant did not state the actual percentage of the ADAs recorded. And furthermore, due to the high incident rates of ADAs, efficacy was significantly reduced, which in our opinion is not desirable. In continuation, next slide. So as an increase in ADA increases loss of response and increase of adverse events, the risk therefore outweighs the benefits of having the drug on the market. And considering that for our country, we have other treatment options, a negative opinion would be given for this product. My colleague will take over from there.
Good afternoon. This is Mfaweli taking over from Prudence. The question that we were asked, the other question we were asked was, what is your opinion on voting in a regulatory process when it comes to efficacy and safety of a medicine? So we Our opinion is that it might cause bias on the decision made, and we strongly felt that voting was not a very good approach, and a decision should be based on sound scientific evidence and assessment of the risk and benefits that concerns of the particular application. Next slide. The next question was present a summary as to whether you would grant a positive opinion for your agency or not based on information available to you. So in this particular study, we noticed that the the subjects did not include African population, and there were some issues on the on the ADAs, and based on that fact that there were issues on the ADAs as provided in the previous slides, and information was not available for the African population, we then came to a conclusion that the safety and efficacy in our ethnic group has not been ascertained, and absence of these studies, we we thought that we would give a negative opinion as an authority because we have an alternative on the market, which is Remicade. The next question. Summarize in one slide what you have learned from this case. What we've learned from this case is that firstly, voting is not a very good approach in making scientific and regulatory decisions, and there's some since there are different views, and some pending issues might have to be addressed at country level. Next slide. So this slide talks to the post authorization documents that we provided. So the applicant provided an update of the RMP. They submitted studies to be able to provide further information on the efficacy, safety, and immunogenicity. Because initially there was a concern on the efficacy of the loss of efficacy due to the ADA development. So in the post authorization documents, this issue was actually addressed by the applicant, where they provided further studies, and they monitored the efficacy, safety, and immunogenicity. Next slide. So that's, as I stated, post authorization. So the data was evaluated, and there was a 78-week extension phase data from the phase 3 pivotal clinical study, and from the company's safety database, and real-world data from post-approval studies. So the results actually did demonstrate that the particular application demonstrated maintenance of the efficacy and safety. So the post-approval, actually, the applicant did address the issues that were there on the immunogenicity, safety, and efficacy. But I think our issue on this particular application is that when it comes to our ethnic group, there is no data that is available for this particular study. This ends our presentation. Thank you.
Yeah, thank you very much for that presentation, which was given by all the team members, which is excellent, and also for your own opinions. And I think that's exactly what we want to raise the discussion and have also a look at your own, encourage you to have an own look at what other agencies have been doing, and then, yeah, make your own decisions. And with this, I hand it over to Lise. for more details and in-depth discussion.
Thank you very much. And thank you also to Group 9 for the nice presentation. And indeed, as Berend already appreciated, also for sharing your own reflection, your thoughts of how you would assess the dossier, how you would handle the specific case. That was much appreciated. I have first a few curiosities. In your presentation, you mentioned that you would more lean towards a negative opinion for this biosimilar candidate for several reasons, but you also mentioned that you saw less of a medical or patient need, let's say, to also have this biosimilar on your market, because you mentioned that you have a treatment alternative for Remicade. So I was just wondering if you could share with us if it's then another biosimilar, for example, or what this alternative treatment is that you have for Remicade in your country.
Sorry, I think if I'm getting you right, you were asking.
You mentioned a
Okay,
few times on the slide that you have a treatment alternative for the originator product, infliximab, on your market, and I was wondering what that product was.
that's Remicade.
Remicade
That,
itself. So you mean we
yes,
have the originator, so we see less need to have a biosimilar alternative.
yes, which we have. I think we're not satisfied with the efficacy and safety in our ethnic group. They did submit data post approval to demonstrate that there's no loss of it's still safe and there's no loss of efficacy. That could arise due to the DAs, but in our particular setting, our ethnic group, they have not given us the information that we need. That's why we're saying that for us to be able to register this particular product, they should give us the information that we need because we have an alternative on the market right now.
Okay, so meaning you have the originator, so you see less need for a biosimilar candidate to also be licensed on the market, especially because you have the originator. And do I understand correctly that for the originator, you also did not have ethnic data, or you did?
Sorry, kindly repeat the question.
So you mean you also mentioned that for the biosimilar, one of the elements why you would not grant it a positive opinion would be that there was no testing in your ethnic population. Was this something that was required for the licensing of Remicade to have this additional testing in your ethnic population?
I think for it, maybe because it was registered way back and being the originator product, it might have not been a requirement for it at the time that it was being registered in our country.
So that can also be a reflection as a regulator to see, okay, what was submitted and assessed at the time that we evaluated and licensed the originator. And then one can also ask themselves the question, do we then want to have additional data or requirements for the biosimilar, as we have already quite a lot of experiences, multiple years of use of the originator in that population with supportive data of efficacy and safety and also that population. So this is also something that you could take into account. I was also wondering, because you nicely presented the divergent opinion that happened for Flixabi in 2016 within the centralized procedure at EMA. And I was wondering, it was at that time 2016. Do you think that today regulators might assess it differently, taking into account that we have already progressed some years and also taking into account what the changing attitude is of regulators towards comparative efficacy testing? Any thoughts on that? And also, of course, considering the fact that there was a lot of post approval data that then further contributed to the overall package of biosimilarity. Any considerations or reflections that you think that regulators might today take a different approach than what they did in 2016?
So I don't know if I got your your question correct, that at least considering the fact that they provided us with more information, that there's been updates on what has post-marketing updates, I think regulators would actually consider giving a positive opinion in that line, because there's new data that is coming up every day and it's seemingly positive.
So the audio quality is not so great for me to understand at the moment, and I think it was Cynthia speaking. So perhaps Cynthia, if you could try again, because I tried, but it was not so easy for me to understand. Thank you.
So I was saying,
This is
considering
better.
that
Thank you.
data that has been brought forward in the recent years, regulators would actually consider giving a positive opinion from the one that was given in 2016.
And do you have some arguments why you think that perhaps they would give a different opinion? And also, just to clarify, at that time there was a divergent opinion, but in the end, overall, by majority, a positive opinion was provided, and also the medicine received a marketing authorization. So from that time already onwards, it received a marketing authorization and was made accessible for use in Europe. So already at that time it was positive, although some members had a divergent opinion. But if you could maybe give some arguments why you think that regulators today might take a different might all have a positive opinion, then that would be useful. Thank you. Any reflections on what the current regulatory thinking is about comparative efficacy testing for biosimilars?
A bit, we have been discussing that last week, no? So it's a bit of a recap of what was being discussed last week.
I hope I got the question correctly. So the question is, what would be our opinion let's say in 2025, what would be our opinion with everything that is there? So I think she had brought out a good point that yes, we do have Remicade on the market, and probably what we'd have to do is, since our major concern was the studies in our ethnic group, what we'd have to do is that we would have to check what studies were provided, and would then consider giving a positive opinion, and then would have to follow up with post-market surveillance. That's our response. Unless my colleagues want to add something.
Thank you, Prudence. I also see a hand raised by Birka, please.
Yes, thank you. I think it's a very interesting and very challenging discussion, what we will see here. When the colleagues asking for ethnic additional studies, and you have Remicade on the markets in several times, do you ever have seen that there are some influences due to ethnic considerations on the efficacy and safety of Remicade to substantiate or to justify your request for additional ethnic studies?
Yes, so as I said earlier, I think she brought out a good point that we have that product on the market, that's Remicade, which they were demonstrating biosimilarity. And if we have it on the market and we do not have any issues on the safety and efficacy, I think we, this is Mfaweli, if we did not see anything post-market surveillance for the original product, then I think our request then would not be justified in this case. That's why I stated that we would have then to give a positive opinion, because we're comparing it to a product that's registered and biosimilarity has already been demonstrated.
Thank you for the clarification.
Thank you, Prudence. Thank you also other students. Any other questions from anyone else in the group?
I have a question not on specifically on the biological issue, but on the comment regarding the voting system. And the background is a bit like that in Europe, obviously, we have many different countries sitting around the table in the regulatory submission discussions. And that might be also a point for discussion then for the future African Medicines Agency. Depends on how the system will be finally set up. But one needs to come to an agreement between different regions, different countries on a certain product. And I just wonder what else can one do if there are differences in opinions on how to interpret some scientific data. And if the voting is not an appropriate process, how any comments on what would be an alternative? Anyone from the team?
It's, um, uh, we would then, I think, even though we thought that the voting system is a bit biased, I think we are also of an opinion that I know there can be different views and different opinions. Then I think there should be then an allowance that the final decision will be at country level. So each country will then have their final decision on the particular product, because I don't see any other way out where you have different opinions.
Regina, any comments from your side on that or another issue? Regina, you raised your hand.
Yes, thank you. And I'm very sorry, I'm struggling with my device. Thank you very much. Good afternoon, everyone. Adding on to the discussion, not about the voting system, but on the biologics and the biosimilar question, approvability, given the lack of data in the very population. I would just like to add to Birka's question, if you don't have any signals from the originator product as yet, when you decide on approvability of the biosimilar, I mean, is it entirely reassuring? The question, because my question isn't this also a question of the quality and of the post-marketing data that you get in your country, and the collection of post-marketing data, whether you see a signal or not? So it's a bit provocative, perhaps, the question, but I think only because one decision has been taken to approve the original biotherapeutic product, does it necessarily mean that for biosimilars there's never an issue? And I'll stop here. Over. Thank you very much.
Answer, because I think we need to close the session now.